My name is Anyou Wang. First, I will thank the committee invited me to present my work here. My work interesting is in computation biology, big data and AI, developing the software and computation algorithm to understand the bigger picture of the bigger biology, bigger data. So one example I saw you today here is the non-coding RNA as a bigger picture for the cancer, and then combine the AI neural network to detect the all cancer type. The title is multiple cancer type, actually the all cancer type. So detect all cancer type with population level can dramatically reduce the mortality rate. In the population level, not all the cancer type, and then no one can do that right now in all cancer. And not only one type of cancer, of course, one type of cancer like breast cancer, you can use the image to screen it, but right now in all cancer, no one can do that. So two primary factors contribute to that barriers. One is the biomarker bias. No endogenous biomarker available for all cancer right now. And then the second is the mathematical model prediction inaccuracy. Right now, the algorithm is not working very well. So to avoid the two things, you increase one of the universal system to detect all cancer. So first thing we need found endogenous biomarker for all cancer type from bigger data, no one set of data for all bigger data. And then the second is using AI, the deep neural network to get a better result for that. So I get one bigger data set over there, and actually it's the biggest data set in the world right now from TCGA database. And they have 11,000 sample of cancer sample, and then 36 cancer type over there. However, some of the cancer type have the sample size less than 100. So I delete this small cancer type of a small sample size of cancer type, and then totally get 26 cancer type over there. And then totally have 9,000 sample, include 8,000 cancer and 600 normal. And then this data set, a bigger data set, so include the all cancer type normally, and then we present all the cancer type I have right now. So the assumption is this bigger data is all represent all the cancer. And then the data available already are how to find endogenous universal biomarker from the cancer sample is very challenging things. First of all, they have a lot of countless confounded variable inside this data set. Whatever data set, they have a lot of confounded variable. You don't know what one. For example, in this data set, the TCGA do an excellent job to measure level epidemiological variable earlier. And then I try to understand how the variable affect the gene expression, the genome. I use the CCA, the correspondent canonical analysis, and to plot it to see it, and then plot as a vector. vector have two property. One is the orientation, the direction, and then if the same direction, and then they contribute move this direction. And if the like the 90 degrees the angle, the direction, maybe it not contribute anything. So one is the direction. Another one is the amount, the strategy. And then here, if the contribute more, and then the line will become longer. The longer line more important. So that's it means that expression sample and then contribute most by the alcohol. Of course, smoking also contribute, but maybe only 0.5% of alcohol. So overall, actually, alcohol is the most important factor to cause the cancer sample expression, actually. Yeah, that's the result after my publication and after about 1 year or a couple months, and another publication also proved that alcohol is the most important from the International Cancer Association and published license. So this proof the alcohol is most important than other factor. Like smoking, other people think is most important, but actually not. The alcohol is most important. The most interesting is the size, this cancer size type, and in fact, they set out the sample, but no significant as alcohol. The most strange that's group of the sample, and then no one knows what exactly contribute to this largest, most important group of there. So it means right now all the variable we think is most important, but actually maybe not. We don't know what one is most important. It could contribute to the cancer bigger picture. So the cancer actually they cause a lot of things. So we don't know what one. In this case, I use the principal analysis to check out how many principal components contribute the variance. And in most of the time, most people think maybe principal principal component 1, 2, 3, that's enough to account for the variance. Actually not. 1, 2, 3 maybe counts like 10% of the, so means nothing variance. So I plug it until 200 principal and get the 0.6% of the variance contribute. So the PCA is independent component. Okay, each PCA independent. So one Nature paper published try to use the individual index like a more than 10 index to try to measure it, to measure this kind of variance. Actually, 10 or 20 this kind of variable index actually don't do so much thing, much better than single index, but not do so much thing, because the 10 index maybe not independent. Even in the independent PCA don't cost so much. So it means so far all the mathematical idea is don't make sense actually. To if you try to measure all the variable variance, it don't make sense. Always bias, always wherever people publish in natural science, wherever share, it doesn't matter. So how can make no bias make compute that kind of what's biomarker come out. Nobody how can do it. So I created one software called iSurvival, and then to include all the PC 200, also include all the that kind of variable, epidemiological variable ODL, and then remove all the confounded variable, and then found 56 non-coding RNA as the endogenous biomarker. And why is the non-coding RNA is important here? They have reason, because the non-coding RNA, first of all, save the energy in the human genome. Like a human genome, why? Human genome have 98% is non-coding, and then only 2% are coding, because it like save the energy. Like the mouse genome have 80% protein similar with the human. However, the mouse only have a life span, only can survive maximum 5 years. The human can survive more than 100 years. How come? Because the human have more long non-coding, the non-coding RNA, or not necessarily long-coding, all the non-coding. That's why the human and they have long non-coding RNA, have non-coding RNA who they are, and they can survive. If the human, all the non-coding become the coding, maybe human can only survive 2 months and will die, never go to 1 year. So the protein coding actually consume a lot of energy. They need a long energy. So the protein actually just work as a backbone for the human genome, and then mode of function is provided by non-coding. So I plug the whole animal kingdom mitochondrial genome range and then non-coding RNA content, and then you see the negative, and it means more non-coding RNA, and they save more energy, can survive longer. This is why non-coding is very important for the human genome. And then more importantly, the non-coding RNA, the coding RNA, how they are distributed. They are unique functional system. They don't depend on the enhancer and polymer A, polymer 2, they kind of protein system. They have their own system, and then they regulate by their own self, and then regulate the bunch of the kind of human genome over there. And then finally, and then non-coding RNA actually endogenous rule the cancer regulate RAM, and then the protein only govern the normal. The normal condition, the protein trans-regulate the genome, and then in the cancer, there are non-coding RNA size regulate the same chromosome, 1 million base pair this area. So that's why the non-coding RNA become the endogenous biomarker for the cancer. A lot of people publish the death protein is the what kind of fundamental mechanism, another one, whatever, and then publish a lot of paper, but actually not that's fundamental mechanism for cancer. Cancer fundamental mechanism is non-coding. Okay, so wherever you can do the mutation for the coding and mRNA, wherever it is, just by for the specific cancer or specific person, you do the precision medicine is okay, and they treat this person as a mutation, but they'll never go to the fundamental mechanism for all cancer. No mutation concerning single cancer type, not only all cancer non-coding RNA, make sure. So after getting non-coding
Dr. Wang, we're almost out of time. 2 minutes. Okay.
Oh, sorry. So after get that, then biomarker, and then build an AI model, 3 land over there, after AI model, and then combine AI model by non-coding RNA, and then you see 13 biomarker get 98%, 93% AUC, and then 50%, 15 biomarker get 96 AUC here, and we are stable, and to tell code and plot in my website compile.org, and then you see ODL from biomarker 1 to 15, and then go up and up again, and 96%. And then if classification, of course, most of people, the model that work on their own data set, but not work on independent data set. And my model work on independent data set. I use a validation tool, independent data set. One is from the International Cancer Association, another was blood exosome sample. And then that's how to the 7 cancer type ODR, 1,000 sample. And then I use the raw data, their TPM data. I don't do anything, just raw data. And then still get 78 AUC there. And then most strictly, and then use the blood exosome data. And then the 12 cancer type. This is cancer type not that match my cancer my cancer type in my model, but still working very well. Only how 7 biomarker match my model, and I use raw data. I don't use any, I don't do anything, and then project my model is working 72% of AUC. So it's working very well actually. And then my model perform the certain model. I like write them first decision tree, and then much more significant over. And then of course individual cancer type you get 99% of AUC, and that's 6 biomarker. As most of them, the 1 biomarker that work well. So summary, and then that's non-coding RNA plus AI model can discriminate all the cancer type, and then 96%. Individual cancer type 99%, and then AI model of course traditional better than traditional model. And then I promise one kind of practical cancer screening system actually maybe work very well for all the population level for all the cancer. Okay, thank you for your attention, and happy to take your question.